Hello, and welcome to HPA Core Module 2 Biology, Lecture 8 on the cardiovascular system. This represents the last of the basic biology segments that we will be instructing before transitioning into the fundamentals of strength training. Please note that these biology segments are intended to be an introduction to the topic, and there are many more advanced topics that you may have questions about that will be derived from these presentations and covered later. Understanding the cardiovascular system is central to understanding the function of the human body. You cannot really process or really analyze a lot of advanced exercise adaptations or even simple things like respiration, cellular respiration, and nutrient processing without a real understanding of the cardiovascular system and how it works. So just a couple of figures on it. As you can see here, human body does contain approximately 5 liters of blood. This is assuming a 70 kilogram human. Now, obviously, larger humans have a greater blood volume and smaller humans have lesser blood volume. There are gender differences as well, but those are due primarily to differences in body size and, of course, body composition. Skeletal muscle tending to have slightly better perfusion than fat. As mentioned down below there, you can see the heart pumps 7,500 liters per day. Again, this very much depends on activity level. It depends on the size of the individual's heart. It can even depend on things like, you're talking about altitude and conditions like that where a greater amount of blood volume is needed to transport the same amount of oxygen. Another critical point here, a single blood cell placed anywhere in the body will take about a minute on average to circulate through the entire system. Now, if that blood cell is going up to the brain or something close to the heart and then circulating back, or even just going to epidermis fairly close to the heart, obviously it'll come back to the heart far more quickly than, say, something going out to your big toe, or blood heading out to, say, connective tissue or into joint capsules down again in your feet. So that's just an average figure. Now, what is remarkable is how small the average stroke volume is. I mean, 70 milliliters. Granted, if you measure it out, it looks fairly significant, but if you think about the amount of blood in your human body, you start to get a sense of just how, I want to say, almost constrained the system is. And though we think of blood as flowing, we almost need to think of it more as kind of pulsing and pushing itself through a very constrained system under pressure. Think about this more as filling a tube with a kind of viscous liquid, and rather than just watching it flow through, think about just kind of forcing it through and putting more and forcing it through. So just to get the sense that the entire circulatory system is, by its very nature, has a lot of passive resistance. In fact, interestingly enough, blood temperature tends to be higher than your quote-unquote core body temperature, simply because of all the friction, resistance, and pressure in the bloodstream. Here is a broken out diagram of the cardiovascular system. Now you can see, of course, the heart is central to this system. You can see how the blood flows through the heart, into the lungs, back into the heart, and out into the body. The most important thing to note here, just as an interesting aside, is that the blood, when it flows away from the heart, obviously it can flow to the upper body, can flow to the lower body. It is important to note that, first of all, the renal system, the kidneys, are kind of their own entity here. If blood passes through the kidneys, it neither comes from nor goes to any other organs first. Now, of course, this seems to initially, at first blush, make sense, but notice that is in complete, that is different than blood that flows to the stomach and intestines. Note that blood that flows to the gut and is therefore responsible for, electrolyte exchange in the gut, or extracting nutrients and possibly, yes, extracting any sort of pathogens or environmental toxins or anything else, goes from the digestive system directly into the liver. Of course, blood can go to the liver separately on its own without going through the digestive system first, but everything that goes into the digestive system and then into the bloodstream passes through the liver before passing anywhere else in the body. And it's important to note. There we go. Blood is composed of three major components. There is the plasma, which is the basic blood volume carrier fluid, red blood cells, and white blood cells. It is important to note that the pH of blood is carefully controlled. Any deviation outside this 7.35 to 7.45 pH is extremely dangerous to the body. Number of reasons for this, as we know, and as we've seen, so many bodily processes are based on maintaining the idea of maintaining a ion gradient, of maintaining a pH, of maintaining polarity in cells is so crucial to operation. Enzymes only respond well and process well at certain pHs. When we look at basic chemistry, we look at the processing and exchange and interconversion of chemicals, and we always note that these are two-way streets. You have your reagents, or you have one side of the equation, and you have what is created on the other. These two species, the chemicals on one side and the chemicals on the other are always in balance. You always have things interconverting between the two. The pH of a system, the acidity of a system, can very much skew things one way or the other. Everything in the body is about homeostasis. You start to modify temperature, you start to modify pH outside a very narrow range, you can get severe complications. If pH goes too high or too low, these are medical emergencies. A pH below about 7.2 or above 7.6 are extremely dangerous. Now, I know we may have a subsection, a breakout section on what we call, acidotic and alkalizing foods and supplements, and we may go a little bit more into what those, what that theory is all about and what these products are claiming. But as you can see, the idea of your body being naturally in a daily acidotic state, for example, just don't hold water. In other words, that is not compatible with life. As I mentioned earlier, blood temperature is slightly above the core temperature, again, due to pressure and friction. Okay, so talking about the blood, the plasma. Now, again, we have three separate components, and of course, if you've ever had a blood test done, and you've ever gotten the chance to see one of those centrifuges, or if you work in medicine, you see those centrifuges, you'll see that the blood separates out into its components. The plasma is the relatively clear liquid in the blood. It is the majority of blood volume. Now, the plasma is everything, effectively, that is not a blood cell of some sort, or a platelet. It contains water, as you can see. It contains proteins, many kinds of proteins, hormones, nutrients. This is basically the catch-all, again, for everything that's not a cell. It also contains gases. It can actually, now your plasma volume can actually change fairly significantly. When you talk about changes in blood volume due to dehydration, due to a lot of other factors, this is all due to your plasma volume. So interesting, again, to note about plasma is that contains albumin, globulins, fibrinogens, all of those. A lot of those are proteins that are responsible for, we're talking about everything from, clotting factors, fibrinogens, response factors. Effectively, just the thing to go with here is that plasma is kind of the everything else catch-all. Now, the other component of the blood, we talk about the red blood cells, or erythrocytes. They are fairly unique-looking cells in the human body. When you look at them, they look like these little indented discs. There's a reason for that, of course. Now, first of all, red blood cells have almost no organelles whatsoever and no nucleus. As we mentioned, they don't even have mitochondria, so their own energy source, the way that they actually get their energy is strictly through anaerobic fermentation, the whole lactate pathway. Now, the reason for this, of course, is their energy needs are fairly low. All they simply do, for the most part, is serve as vessels that contain hemoglobin and transport oxygen. Now, they have a 120-day life cycle, so they're cycled around. I mean, that's actually pretty long-lived. A lot of white blood cells, for example, don't last nearly that long. Now, interesting properties, again, of red blood cells. That particular shape of theirs, this kind of disc shape, when they move through fairly small capillaries in the body, for example, those capillaries may be narrower than the diameter of a single blood cell. These red blood cells can actually fold and kind of curl up like tacos or burritos and kind of work their way through. So these are very pliable, very small, very specific, highly specialized cells. Now, you can see hemoglobin on the right. Now, remember the discussion on proteins? Each one of these chains, these beta and alpha chains, are basically forms of this protein, very arranged in a way where you notice that the structure of these proteins, the function of these proteins, notice the Fe2+, which is the iron group, the iron ion attached to the heme group. That is the site where oxygen is bound. A very interesting property of hemoglobin, and something we will discuss when we talk about exercise adaptations, altitude training, certain supplements, and even the acute response to exercise, is how this hemoglobin protein, this four-lobed protein, can change shape and change configuration based on the presence of other molecules, and either expose themselves to more oxygen binding sites or to fewer, which can change how quickly and how readily those iron groups let go of oxygen. Now, why would this be crucial? Well, when the body is under severe hypoxic stress, either during exercise or at altitude or anything else, what starts to happen is, and this is something we'll go into later, something called the Bohr effect, where the sudden change in pH associated by kind of the buildup of carbon dioxide, low levels of O2, etc., that pH change in the blood can actually change the configuration here and make these cells more likely to release oxygen to working muscles, which makes sense if you think about it. If the body is under stress and needs more oxygen for function, then hemoglobin is more likely to release larger amounts of oxygen from the molecule itself. Because remember, hemoglobin molecules, red blood cells, don't lose all their oxygen when they go through capillaries. A certain amount of oxygen still comes back. These are not perfect shuttles. They don't go out to the capillaries and dump everything off. They dump off a certain percentage, and that percentage increases, of course, due to certain environmental stressors. Anyway, hemoglobin itself, at the end of this 120-day life cycle, the red blood cells are typically kind of nearing their end, and the bone marrow, liver, and spleen all have regions that break these blood cells down and reharvest the hemoglobin. Hemoglobin is very interesting when it's broken down. Some of the amino acids can be reused to make more hemoglobin. Other compounds like bilirubin, etc., are basically taken, and these colorful portions of hemoglobin themselves are incorporated into other things like bile, bilirubin, bile. What's interesting about this as well is note that these are very colorful portions of hemoglobin that can be green and yellowish in color. When you get a bruise, it is the breakdown of hemoglobin from red blood cells that are dying or have ruptured in that area that are responsible for those green and yellow colors. So that yellow color you see in a bruise is actually one of the same compounds that then goes into the liver and is used to produce bile. Fun fact. So that is it for red blood cells. Now, white blood cells. It is important to note that white blood cells represent a massive category of cells. So many here that we will not even attempt to scratch the surface. If we want to do a separate section on immunology, we could easily teach entire graduate courses of study just on the various categories of white blood cells or leukocytes. They are basically, all you need to know is they are basically divided into two categories, which have to do with, honestly, the way they determine these categories was with the presence of little granules in the cells when observed under certain kinds of stain, granular and agranular. Now, you can see that the granular cells tend to be more associated with what we call nonspecific or innate resistance. All of these cells, whether granular or agranular, can be responsible for identifying pathogens, destroying pathogens. In many cases, like I said, a lot of monocytes, lymphocytes, you have cells here that that can specifically target certain pathogens and tag them, etc. For the most part, the granular cells tend to be more on the innate defense / non-specific side, whereas most of the defensive leukocytes, that are responsible for identifying previous antigens, like in an established immune response, are going to be of the agranular sort. Now, this is just a kind of a general categorization, tremendously simplified. We may discuss this a little bit later in separate breakout segments. Important to note, unlike red blood cells, these white blood cells can leave the circulatory system whenever needed. They can kind of push their way through blood, the capillaries, and get to sites of injury, get to sites of infection, kind of move their way around the system. Now, of course, these are then extracted and end up in the lymphatic system after either absorbing or releasing contents. But that's a separate section, of course. So another thing that's important to note is that when these cells are released to certain areas, they do not essentially transport themselves across the barriers, across capillary walls or across vessel walls at will. We notice that when we talk about leukocytes, we talk about a lot of these pro-inflammatory cells. The inflammation response, which we will discuss pretty extensively during the rest of this course, also has an interesting property where blood vessel permeability is related to the presence of certain quote-unquote pro-inflammatory compounds. When we talk about pro-inflammatory compounds, we talk about vesicle permeability, what we're basically saying is that if the body detects some sort of injury or insult or anything else, it is in the body's best interest to increase the permeability of blood vessels to allow for greater number of leukocytes to get to the target area. Now, a lot of these leukocytes, not only are these compounds that are responsible for mounting an immune response, but many of them are responsible for actually releasing growth factors and repair factors to a site. So if there's an injury, the first white blood cells that go there are a lot of these pro-inflammatory cells. When these pro-inflammatory cells get there, they release signaling factors that cause blood vessels to become more permeable, which allows other growth factors and other white blood cells to get there. So again, something we will discuss in much greater detail later, but please note that when we talk about pro-inflammatory responses, this is one of those chief mechanisms that's always worth understanding. So the last component of the blood would be platelets. Now, platelets are not actually cells. They are fragments of much larger cells, basically fragments of the cytoplasm that are broken off, and they have a couple different functions. They release growth factors as well, and they also aid in clotting. We'll discuss these more later when we discuss injury. The heart, the center of the cardiovascular system. I'm going to leave this up just for a moment so we can take a look at the structure. Now, of course, when we discuss endurance training and we discuss more in the cardiovascular adaptation, we will discuss more in the heart, and specifically cardiac muscle adaptations, certain issues with the heart and structural defects that are frequently exposed by exercise, etc., etc. But all that you really need to note here are the presence of the right atrium, left ventricle, left ventricle, and the rough flow of blood in the heart, which we'll look at in just a moment. A few more stats on the heart. 10 million liters of blood pumped per year. The heart is a very, very busy organ. Now, cardiac muscle, the type of muscle in the heart itself, it is a contractile muscle. It is not smooth muscle like what is found in the intestines, for example. It is still considered a type of striated muscle. Now, when I say arranged in a contiguous fashion, a lot of cardiac muscle, if you look at the heart itself, it is almost effectively one large muscle with the muscle fibers kind of arranging themselves in almost a contiguous whorl pattern around the heart. And there's a very specific reason for that. Cardiac muscle cells, what makes them different from most skeletal muscle is that, first of all, they tend to be very, very short. These are very short muscle fibers themselves, very short muscle cells. And they only have a single nucleus each. Now, this makes sense, of course, because we talk about multiple nuclei in skeletal muscle. Part of the reason is to allow for greater hypertrophy, larger nuclear domains, etc., etc. This is obviously not needed in the heart. This is not a muscle that can undergo hypertrophy to the same extent as skeletal muscle. Of course, it can certainly undergo some. Now, the way cardiac muscle is arranged, these muscle fibers aren't simply attached to one another. They have these intercalated discs in between them. If we look at these discs, and we can discuss a little more in the specific structure and function of these discs, these are basically the surface, the domains between these muscle fibers that allow contraction to kind of propagate through them. And we'll talk a little bit more about the properties of that later when we give some advanced segments on HRV and the nervous system and response to changes in heart rate. So it is important to understand, of course, that these discs are also kind of anchor points for cardiac muscle, but are not, anywhere near as robust or durable. These are not tendinous segments or anything else. Think of them more just as cellular regions. Interesting fact about cardiac muscle, most of them are capable of depolarizing themselves. If you take cardiac muscle and put it in a petri dish, it will actually pulse very, very slightly on its own. Now, there are a certain number of cells in the heart that are still these cardiomyocytes, these cardiac muscle cells, that actually generate impulses themselves, that make the pacemaker system of the heart. Now, if every cardiac muscle cell can effectively contract on its own a little bit, what would happen if there was no central pacemaker is that the heart would just sit there quivering, and all the individual muscle fibers would kind of quiver and twitch and everything else. When we look at a heart that is quote-unquote stopped, and we start to look at ventricular fibrillation, atrial fibrillation, well, let's talk primarily about ventricular fibrillation. What we're talking about in that case is a bunch of muscle cells that are contracting without any sort of central pacemaker or central governor. And of course, the heart just sits there twitching and releasing numerous electrical impulses. So the whole, flatline concept, and many of you who are in medicine and medical professionals, of course, know this, the whole idea of no electrical activity at all and no contractile activity at all is very, very rare. What is far more common is that a lot of these cardiomyocytes are just kind of twitching and contracting on their own with no central governor. Now, we'll talk about the governor, the pacemaker systems in just a moment. Interesting fact about the heart. If the pacemaker systems, without the input of any other nervous systems or any of the autonomic nervous system in the body, the heart would beat at 100 beats per minute. That is almost regardless of anything else. It is the autonomic nervous system that actually governs the heart rate and changes a lot of your resting heart rate. And it does that in response to numerous feedbacks, which we'll talk about in just a moment. Now, the SA to AV bundle of His, that is the particular nodes, and we will talk about that in just a moment. But please just note that the SA node is the sinoatrial node, the AV node is the atrioventricular node, and the bundle of His is sort of a cluster, a pacemaker cluster, which we'll see on the next slide. So, if you look on the right side of the screen here, now look at figure 1, now where it says start. In the very, very upper left, moving from 1 into 2, you'll see that the SA node is a little yellow node at the very top of the right atria. Now notice that since we're looking from above, the right is on your left and the left is on your right. Don't ask. So notice how this is where this signal starts, is from this SA node. And moving from 2 into 3, you can see the wave contraction of electrical activity in the heart, which causes all those cardiomyocytes to gradually contract one after the other. And then in 4, right there, you can see how the AV node is activated by this propagating signal. And then the AV node, through its own system of further activation, then tries to basically, rather than having the signal propagate and start kind of at the top of the ventricle, notice how the signal travels preferentially faster through and causes contraction kind of from the bottom up, moving into 5 and 6. The reason for this, of course, is to kind of optimize blood flow and optimize how blood moves around the heart, so the whole thing isn't just squeezing in one single wave type motion. So the proper operation of the SA to AV node and that conduction is very much responsible for a properly operating, efficiently operating heart. But what actually controls the heart rate? We talked about that just a little bit earlier. The SA node is kind of your control center, but of course, if left to its own devices, it'll just pump everything at 100 beats per minute. That doesn't really do much good. So the autonomic system, autonomic nervous system, as we mentioned, both sympathetic and parasympathetic, are responsible in kind of this interplay, this back and forth for controlling heart rate. So the sympathetic nervous system releases norepinephrine. What norepinephrine does is it speeds repolarization of muscle cells. Now remember, muscular contraction, when we talk about the actual ability of a muscle to contract, we're talking about that repolarization and depolarization and ions going back and forth across the barrier, which then leads to the influx of calcium, which causes the actin and myosin heads to crawl across each other. If you speed the rate of repolarization, what you're doing is you're effectively getting the muscle cell ready to contract much, much sooner. So if you have a greater sympathetic response, the autonomic nervous system is releasing a lot of norepinephrine, and remember where that was generated in the adrenal glands. If norepinephrine starts propagating into the system and causes faster repolarization of muscle cells, you're going to speed up the rate of spontaneous contraction, which applies to, of course, all the muscles in the heart, or all the cells in the heart. The parasympathetic system releases acetylcholine. What that does is it opens potassium channels that are otherwise gated and slows depolarization. Now remember when we were talking about the cell membrane and different types of channels and how potassium channels tend to have a gate on them that, when you close that potassium gate, what you do is you start building up potassium ions inside the muscle cell. What happens is then that leads to development of that, kind of electrochemical gradient, that polarization. If acetylcholine basically lets those gates stay slightly open, doesn't let them all snap shut, that's going to slow the depolarization. Again, kind of slow the actual spontaneous exchange of ions that happens when a cell tries to depolarize and tries to contract. So notice that these two systems can both operate at once. If you speed the repolarization but slow the depolarization simultaneously, depending on the amount of norepinephrine versus the amount of acetylcholine, you may still be able to have one kind of overwhelm the other. And the reason why this is important is to note that this is not an on or off switch. There is always parasympathetic activity. There's always acetylcholine being released that is maintaining this, that is slowing this depolarization. There's always a little bit of norepinephrine in the system that is speeding repolarization. These two different systems are always in a balance. So it is, again, carefully controlled by arousal levels, and you can always see that since most of our heart rates are not at 100 beats per minute, there's always a slight parasympathetic dominance in the system, as a way of speaking. Of course, many, many other things control heart rate as well. We talk about baroreceptors, which these are parts of the heart that sense blood volume, proprioceptors, which detect the amount of activity level, chemoreceptors, which detect blood gases, temperature, stimulants, depressants. All these other things can have an impact on the rate of contraction and contractility and have some effect on how quickly that node is firing. Again, what is interesting here is that at no point, can we say that any one system dominates. So, for example, from a practical level, when we talk about using heart rate to measure calories burned or measure, activity level or anything else, please note how many different things here can be affecting the heart rate outside of, let's say, the proprioceptors. When we think about, an athlete is running. If we think about, the heart's speeding up because the muscles need more oxygen. So what we're talking about there is probably the proprioceptors and the chemoreceptors detecting lower amounts of circulating oxygen. We're talking about blood gas asymmetries, etc., which may be causing the heart rate to speed up. But notice how many other systems may be at play here, which are also activated concurrently. What about changes in blood volume? What about the presence of depressants? What about excess in sympathetic tone or sympathetic arousal, or even very low level of sympathetic arousal may all be contributing here or not contributing to a change in heart rate, which may skew your expectations. So heart rate, when it is associated with certain types of exercise, is kind of a proxy for the overall state of the body at any given moment. When we talk about calorie burn during exercise or anything else, or heart rate as a level of intensity, it is not always an exact science. What the heart rate is basically saying is, okay, with all of these factors, all of these things going on at once, what is the approximate level of arousal and output and expenditure of this individual? It is not an exact science because there are so many variables involved. And that is the most important thing to note. They are all always involved. The degree of which is what matters. Last point here. The amount of output, the amount that the heart pumps out in a single beat, is known as the stroke volume. That is not the entire volume of the left ventricle. That is the amount that is actually pumped out. Stroke volume times heart rate is total output. This is something we'll come back to later. It is just important to note that stroke volume, when your heart beats, it obviously doesn't get rid of all the blood in the left ventricle. There's always a little residual left behind. There's potential for various heart conditions with various amounts of backflow, improper valve closure, etc. We will come back to that. So, on heart rate, let's take that and discuss an interesting topic that has been around for, at least since the 90s, but has recently come into a little more, recently had a lot more attention, shall we say. Heart rate variability. What is heart rate variability? Well, those of you in the medical field may notice that this on the left is the typical EKG, or electrocardiogram. The K comes from the German for cardio. Now, you notice that the different waves, if you've ever been curious what they represent on an EKG, look at the graph on the right. No, look at the figure on the right. Now, remember how we talked about the signal propagating? Each one of these steps represents a certain level of, kind of electrical activity in the heart. So we can see the different polarization and kind of repolarization as we go along. And I'll let you look at that just for a moment. Now, what I want you to really pay attention to here is that R stage, that R interval, which actually represents a coherent burst where a signal is propagating from the AV node through the bundle of His all around the ventricles, which represents a very distinct electronic, or kind of a very distinct electronic signature, which makes a very, very sharp peak. That interval between the two R's, if taken over a period of time, represents your heart rate. That is it is the easiest to note, electrical pulse and corresponds to your heart rate. Now, the interval between two heartbeats, the interval between three heartbeats, you're going to notice that the interval between that R in every single beat is not going to be identical. There is very slight variability in that heartbeat. Look at that EKG strip at the very top left, where you have the graph background. Each one of those top spikes, the top of those R segments, is a slightly different distance or duration apart. The interval between those R's, that RR interval, that variation in RR intervals is what's typically called heart rate variability. It is a very, very normal thing. There are so many factors that can affect your beat-to-beat heart rate. One of the most common and the best known and best documented is called RSA, respiratory sinus arrhythmia. Now, if you think about the heart, and let's go back a little bit to what affects heart rate, think about the fact that your heart is currently in your chest cavity. What happens every time you inhale or you exhale? You're changing the pressure in your chest cavity. That change in pressure is going to change the amount of blood, the amount of venous blood flowing in. You're either going to increase the pressure in your chest cavity or decrease it. That is going to have very slight impacts on how much blood flows into your heart. Those baroreceptors in your heart are therefore going to potentially very slightly change your heart rate for that precise moment. So RSA, respiratory sinus arrhythmia, is something that's very well documented. It's why your heart rate speeds up when you inhale and decreases when you exhale. Interestingly enough, you have greater RSA, in other words, greater arrhythmia, greater variation in younger people and in athletes. One of the reasons for that, I mean, there could be a number of reasons. It could be strength of your respiratory muscles. It could be that these baroreceptors are more finely tuned and more responsive in healthy young individuals, etc. So that greater HRV, that heart rate variability as a result of this respiratory sinus arrhythmia, is actually generally a good thing. I think one of the more confusing things about HRV is people think, oh, well, if there's a lot of variability in my heart rate, isn't that bad? Like, shouldn't it be steady and consistent? That's not necessarily the case. So we will get into an entire segment on HRV later, and we will discuss at great length a lot of the research that goes into it. However, what is important to note is that right now we are aware of dozens and dozens of factors that can all subtly change your heart rate variability. And they change your heart rate variability in certain patterns. These patterns of HRV oscillation, which is expressed in hertz. You don't really need to worry about this. But just note that if you're reading on HRV and you hear high frequency HRV is associated with sympathetic, high frequency is associated with parasympathetic and low frequency is associated with sympathetic, what they're referring to are patterns of HRV oscillation. In other words, patterns of interval changes in that RR interval that are associated with the predominance of certain systems. A lot of times in medicine, HRV is used to assess things like recovery from heart attacks. They are looked at, when we look at things like sleep apnea, we'll see certain patterns of heart rate variability. So heart rate variability has been noted to have a little bit of, it's kind of a lagging indicator, or let's just say a derived indicator of certain states and certain illnesses and certain injuries and other conditions in the body. There is no simple way of saying that, X input causes Y change in a heart rate variability. We are simply saying we've seen these patterns in HRV changes, and we know that these are found in conjunction with these conditions here, and therefore we're starting to create patterns where we're associating this change in HRV with this particular condition. It is recently being used now with the proliferation of a lot of pulse sensing, chest straps, heart rate measuring devices, especially daily wearables. It is starting to be used for overall readiness and exercise and training. The challenging thing here, of course, first of all, is actual quality of data. Original HRV data, this change in RR interval is very, very, very small. It is a very, very small difference between beats that needs to be measured. Certain devices are probably not accurate enough to detect minor changes. As it stands now, as of the current date of this presentation, and we're talking about late winter, early spring 2019, optical devices and wearables do not seem to have, and again this could change, do not seem to have the level of precision that is needed to actually analyze a lot of minor changes in heart rate variability. What you really need for that is any sort of direct measure of electrical activity that is sensitive enough to detect millisecond differences in polarization and depolarization. Assuming you have those, of course, you need to have fairly consistent, fairly constant readings. In a medical setting, when HRV is calculated, you're looking at EKGs that are being left on patients for extended periods of time, and the data is being collected. We are only now approaching the level where people will wear these wearable devices for long enough to really collect good amounts of data. Now, that's not to say optical devices are useless for HRV. There certainly seem to be certain larger patterns that can be predicted from it, but this is still very much a work in progress. So that is a little side on HRV. The last thing to note about the cardiovascular system is, of course, a few properties and, of course, the size of vessels as things come out from the heart. All you need to know from this is that, as you can see, blood flows from the elastic, which are the largest arteries, to the muscular arteries, arterioles, capillaries, venules, veins, and up to the vena cava. Now, this is, again, most of you may remember this from biology class. It's just important to note that obviously the smallest diameter vessels actually have the largest total cross-sectional area. Just to give you an idea of how many capillaries there are in the human body, it is also worth knowing that blood moves extremely slowly through the capillaries, which, of course, if we think about vessel diameter, makes a heck of a lot of sense. And, of course, the difference in blood pressure. Now, please note, again, everything going through the arteries, the blood going through the arteries is effectively getting pumped through. That is why arteries tend to be thicker, have thicker walls. They are under active pressure from the heart and the pump pushing the blood through. Once it gets through the capillaries, it's kind of pulled through the capillaries by something ironically called capillary action, and then dumped into the veins. And from the veins, the primary way, veins in the upper body, of course, get the blood back to the heart via effectively gravity. Veins from the rest of the body pump back to the heart primarily through skeletal muscular action. So the actual active contraction of muscles is responsible for pumping blood back to the heart. Now, of course, pressure gradients, a certain amount of negative pressure coming from the right atria contracting, etc., aids in blood flow. But this is also why if you sit still for any period of time, you start to get swelling, you start to get blood pooling. And if you've been standing for too long and haven't been contracting anything, motionless, especially if you have low blood volume, you may notice that you start to feel faint as the amount of blood returning to the heart decreases. Those baroreceptors notice this decrease, you may get an increase in heart rate and then a decrease in heart rate, and you may pass out. That is all you need to know on the basic introduction to the cardiovascular system. We will, of course, be exploring these concepts in much, much greater detail when we get to various segments on strength training and endurance training.